how was this vaccine able to be developed so fast? And so there were a few things that happened. Um, Proved this vaccine production on the fast track route. Phase one and phase two, which shortened. Um, and also, it was fast because this is different from any other vaccine that's done before, and I'll explain later. But the whole genome of the virus was released as of January 2020, so just as soon as it began, and it was shared with all the research institutions worldwide. And so instead of, just trying to create an inactivated vaccine by trying to grow the virus in a different vector or trying to inactivate it or trying to attenuate it, they're actually targeting a specific portion of the virus to create an immunogenic response. And also, it's a focus on an mRNA vaccine, which are easier and faster to develop. So these are the type of vaccines, the ones that we're used to, usually inactivated vaccines and live attenuated vaccines. Inactivated vaccines are just vaccines that's, of the viruses that's been chemically inactivated. Live attenuated is basically a live virus, but then the genome has been changed so that it's not virulent. And then I'm not going to talk about these ones. Vector vaccines, that's actually the AstraZeneca one, where they actually put the mRNA and whatnot in a different virus, like for the AstraZeneca one, they're putting it in chimpanzee adenovirus, and so you're still infecting someone with the virus, albeit although the virus is not, it doesn't cause sick illness. And the DNA vaccines are one thing, and our focus is on RNA vaccines, which is the majority of the vaccines. And RNA vaccines the technology has been there for several years. More than 10 years actually, and it's the crux of the coronavirus vaccines like the MERS-CoV from the Middle East, and then and then I believe Ebola too. They were all kind of RNA vaccines, but they're not really widely used. And so the RNA vaccine is they're specifically targeting the spike protein of the COVID-19 virus. And the spike protein is on the cell surface of the virus, and it's used to attach to the ACE2 receptor on our host cells to allow viral entry. And this spike protein is relatively conserved, as in it does not mutate, and it's quite also, conserved among the different coronaviruses as well. This is a very important target, yeah. And so they basically knew already the mRNA sequence and the genome sequence to the spike protein, and so that's what they're doing. So what's happening is that this vaccine is actually very simple, as opposed to previous vaccines, which actually contain a live virus. All this vaccine contains is an mRNA that of the virus that would produce the spike protein in our cells. So basically, the vaccine contains mRNA. It's injected. The mRNA is very special because the reason why mRNA vaccines have not been used in the past is because they degrade so easily, and also like it's hard to get into the cell, right? The cell's not going to accept any mRNA. And the other issue is that mRNA can cause inflammatory responses. Our body recognizes it, and they're like, oh, this is not mine. Boom, anaphylactic reaction, whatever. So what they've been able to do for this vaccine that made it possible is that, one, they were able to coat this mRNA molecule in a lipid nanoparticle. This basically protects the mRNA and allows it to be easily absorbed in the cell membrane. So that's one thing. The other thing that they did was that they actually changed the mRNA sequence to allow it to be less immunogenic, to reduce the inflammatory response and boost the translational response. So once the RNA, and remember that RNA is used in the cell to create proteins, right? DNA makes RNA and mRNA, and mRNA makes proteins that basically, for a million reasons, purposes. The mRNA goes in the cell, and then they use our own ribosome to translate the mRNA into protein and spike protein. And the spike protein, gets shed, and also it's within the cell. And what's really cool about this vaccine that's different from other vaccines is that inactivated viruses and live viruses actually don't necessarily enter the cell, so it's going to only be on the outside. So they usually lead to only an antibody response. This is cool because not only will you get an antibody response to the spike proteins that our B cell response, but because the spike protein is also within the cell itself, it actually activates the CD8 lymphocyte response. So not only are you getting antibodies, you're actually getting primed CD8 T cells. And so you have double the amount of response in case the coronavirus happens to enter your system later, as opposed to most, in flu vaccine where you can get an antibody response, you also get a memory T cell response, which is how this vaccine is powerful. I think we talked about that CD8 T cell response, mRNA. Oh yeah, and then people are like, oh, what happens to the mRNA? Well, the mRNA actually gets degraded by the normal cellular processes once it's been translated, and the lifetime of an mRNA molecule is like a few minutes to a few days. So after the vaccine, after the mRNA is done making those spike proteins to let the body know that that's what the virus looks like, none of the genetic material actually remains in your body. So that's what it happens. It's all, and the reason why you use mRNA to make the spike protein as opposed to injecting the body with just the spike protein is because the spike protein is really hard to make outside. So why not just use our cellular mechanisms? That's the idea behind it. Issues with mRNA vaccines, I talked about this already, but ordinary mRNA only produces low levels of protein, and so this mRNA, the nucleoside sequence has been changed so that it produces more. It degrades too rapidly. That's been solved with the nanoparticles, and it can trigger severe inflammatory responses, and that's been changed by also nucleoside modifications to reduce that inflammatory response. So the three main things, the vaccines that we have now are the BioNTech and Pfizer one. It's Pfizer that's doing all of it. BioNTech's just kind of sponsoring Moderna, which is also an mRNA vaccine, and then this one that I can pronounce. But that's one is an AstraZeneca vaccine, and that one's a vector vaccine. And so the other article that every one of you guys have been seeing is, the NEJM article, which is the phase 3 trial. And this is a randomized double-blind study, several institutions, but mostly based in the U.S., enrolling 43,000 participants. And it's split between 20,000. Half of them received the vaccine, and half of them received the placebo. And the vaccine was given at day 0 and day 21. And so, just a overview of the vaccine. It's an mRNA vaccine. Okay, it's also been approved. So there's 2 doses required for this vaccine, and I'll talk later why. And they're given 21 days apart, so about 3 weeks. So 43,000 received the vaccine. It's only 16-year-olds, and then 21,000, 43,000 in the trial, 21,000 received the vaccine. And the efficacy was measured. The 95% that you guys keep hearing is measured after the second dose. And so 7 days after the second dose, they measure that, and then they also keep following it for about 2 months afterwards to see how many people got COVID. So after the second vaccination, out of the 40,000 people, okay, who placebo or the vaccine, 170 people got COVID. Out of the 170 people, 8 of them were in the vaccine group, and 162 were in the placebo. That leads to an efficacy of about 95%. It's interesting, though, because right after the first dose, they also kind of tabulated how many people, got COVID. And, the efficacy was not really calculated per se, but looking at the numbers of people who got it, I think it was like 100 people in the placebo group and like 50 people in the vaccine group, something like that. So that leads to about a 50% efficacy. So even after the first dose, there's already some immunogenic response, but you need 2 doses. And then this study was done, the patients were monitored for safety data for 2 months. It is still going on. They're still monitoring it because usually phase 3 trials, they monitor the patients for 2 years. but this one is for 2 months. The vaccine has to be stored at negative 70 degrees Celsius, sub-zero temperatures. That's storage, but if you're planning to give it, then it only needs to be refrigerated. But if once you refrigerate it, you need to give it within 5 days. And so I'm not sure if Kaiser we have a fancy refrigerator, but then at least if we're planning to distribute it within the next 5 days, it doesn't have to be stored in this temperature. So I think that everyone's interested in what are the side effects, adverse effects. So the adverse effects for this vaccine, there was about fever in about 16% of people, headache 26%, chills 18%, fatigue 38%. I didn't put the slide in here, but compared to the flu vaccine, it's actually about equivalent. Cost $39. It's actually pretty cheap. Excludes children, adolescents, and then pregnant. And so this is actually taken right out of the phase 1/2 study, and then actually no, it's not. It's not this article. This is actually I took it directly from the FDA briefing on the safety data, and you guys can read it later or whatnot. But then as we talked about, the most common were just adverse reactions were injection site reactions. As far as I know, and I looked deep in it, there was no one that had an anaphylactic reaction to it. Lymphadenopathy occurred as well, enlargement of lymph nodes. I don't know about this one, Bell's palsy in 4 vaccine recipients, but then they said that the Bell's palsy in 4 vaccine participants is not that surprising because that's about the same frequency of Bell's palsy in the general population too. So I'm not sure about that. So deaths. So in the whole trial, there were 6 total deaths out of the 43,000 enrolled participants. And out of the 6 deaths, 4 occurred in the placebo arm, and then I think one of them had an MI, and then 2 others were unknown. 2 died from the vaccine side, and they said this is not necessarily from the vaccine. None of these were directly from the vaccine. It's just kind of the baseline rate of death in a population. And the 2 people that died who received the vaccine, one actually died from cardiac arrest 62 days later, so I don't think it's related. It's probably an MI. The other one was kind of a little bit strange. It's 3 days later, but I don't know. The cause of death was labeled as arteriosclerosis, so I don't know what that means, but that was the only other one, so that's weird. But then at least the study people said that none of it was directly related to the vaccine itself. So this was kind of it. Another fun, interesting thing, it's not fun, but it's interesting thing I was kind of digging through the FDA that people don't talk about. There was appendicitis observed in the vaccine participants, about 8 compared to 4 in the placebo. Not sure why, but then I mean, this vaccine causes a bit of lymphadenopathy, and then the appendix does contain, lymphatic tissue. So if that's potentially something, it could just be, chance. It's only 8, so I'm not sure about this causality, but I just wanted to share it with you guys to make you guys aware. So the key takeaways for the vaccine, mostly in the 2 months that's been studied, and also actually it's been studied since February, right? Because there's phase 1 and phase 2 trials, but most of it are just really local reactions that we expect of any vaccine: the pain, swelling, redness, some systemic reactions as well: fever, headache, myalgias, much higher at the second dose than the first dose, and then most were mild to moderate and resolved rapidly. The vaccine did show early protection 12 days after the first dose and 7 days after the second dose, with a 95% efficacy. And safety and efficacy was studied only for 2 months, while in the usual, trials, these things would be monitored for several years. But unfortunately, we don't have the benefit of monitoring a vaccine for years before we get it because of our pandemic. And also, one unknown question is, it's uncertain whether the vaccines protect against asymptomatic infection and finished transmission. That we don't know, right? Because we only have a 2-month data. We haven't explored it. And then FAQ. Okay, so a few different questions that people ask. Okay, how long will the immunity last? We don't know. We don't know how long it will last. Usually typical flu vaccine, the immunity lasts for about a year. This is different though. This is an mRNA vaccine, and then it's specifically priming our body to recognize the spike protein. So it could potentially be longer, but we don't know at least. But at least from the studies, at least for 3, 4, or 5 months, we will have good immunity. Um, and how about people who have already had COVID-19? That is also uncertain. I think the COVID patients, it's kind of weird because the article did talk about a few patients who had COVID that were in the study, but then they weren't actually tabulated into the final efficacy rate, so they were excluded. But some patients who's had histories of COVID, um, they were given the vaccine too. And Dr. Jactiani was saying that patients who've had COVID-19 should get the shot, and there's a reason behind that. So like I mentioned earlier, the reason I mentioned that this vaccine causes not only a B cell response and antibody production, but also a CD8 T cell, killer T cell lymphocyte response, you get two, right? You get the humoral immunity and you get the CD8 immunity from this vaccine. Patients who've had COVID, we don't know for sure, but the theory is that they may not have developed a CD8 T cell response because the virus might not have entered the cell. And so you might have some humoral immunity, but not necessarily, right? And also, if you think about it, this vaccine, it takes 2 doses to create the 95% efficacy. So unless you got COVID twice, potentially, we don't know how much immunity you will get. Dr. Chatiani said once they get past the recovery period in quarantine, we have no idea what the durable response of COVID infection is. That's correct. And then also a few other questions. How about children and women in pregnancy? So this vaccine did not study children and women. It's greater than 16 years old, and no pregnant women were included. They are doing additional studies on these populations, but at least for this current vaccine, it doesn't include these populations. That doesn't mean that it's not safe for them, but it's not been studied, so we don't know. The only thing that we know that is that in terms of the side effects, the younger patients tended to have a more robust side effect, response in terms of the local side reactions compared to older individuals. Also, one person asked, will the vaccine cause false positive results for future COVID tests? And I don't know if they tested it, but logically speaking, it wouldn't, because the COVID test is a PCR test that specifically looks for the nucleic acid of the virus. This vaccine has mRNA, yes, but then it degrades, right? And then it's producing the spike protein. And so this vaccine shouldn't cause false positive results in patients who get it, because this is different, right, from the typical flu vaccine, which is an inactivated or attenuated vaccine. This is just an mRNA vaccine. And then Dr. Lee said, I heard the FDA commissioner said that those who recovered from COVID-19 should wait for 3 months before getting the shot. Okay, yeah, so so that's one can thank you, Dr. Lee, and thank you, Dr. Jatiani, as well. I think that's it. Yeah. Personally, I was on the fence about it, but I think the risk of and the devastation that can come out of a COVID infection, especially with all of us being in the front lines, far outweighs the little risk that this vaccine may present. I think I don't expect, some people also mentioned they're worried about long-term effects. I mean, this mRNA doesn't stay in the body forever, right? It degrades. So technically this vaccine kind of disappears, except the spike proteins produce a little bit, and even the spike protein should be, degraded by the body once the immunogenic response is done. So, and as far as I know, we don't have spike proteins in our cells, so I don't think it could lead to autoimmunity either. Shreyas said, there's a new strain of coronavirus in the UK. Was this made for a spike protein novel to the current strain, or that common or unknown? No worries if unknown. I don't know about this new strain, but like I mentioned earlier, yeah, thanks, Dr. Jatiani. Yeah, we're having a larger grand rounds on this topic again in January that Dr. Jatiani is presenting. And so, yeah, thank you so much. Yeah, in terms of the spike protein, when in my reading, I was seeing that the spike protein, like I said, is a relatively conserved um, sequence in protein. It's something that's vital to the coronaviruses and specifically to COVID-19. And so even among the different variations of the COVID-19, this one protein was the most stable in terms of having the least or none, I think, genetic variation. And so if even if there's a new strain, I don't think it would be robust enough to produce that a different spike protein. Amna said, so if we get vaccinated and then develop fevers, should we still get tested for COVID since those are COVID symptoms? Well, I mean, if you get vaccinated and you immediately have these symptoms, then I think you could assume that this is a reaction. And then I mean, if I had that, I would just probably just wait for like a day or two and see if it resolves on its own. And, and then also in addition to fevers, chills, I think I would be looking for, cough, shortness of breath, and other symptoms because then those would tell me if it's an active infection rather than a reaction to the vaccine. So I don't think I would get tested unless like more of the, symptoms were there. Oh, the last one, yeah, it won't cause false positives. Yeah, it won't. Yeah, because there's no genetic material in the that remains in the body. Yeah. Any other questions, guys? Was this helpful? I don't know. I went through it really fast, but I hope I covered as much as I can. Yeah, I've been looking at this data for like 2 weeks now, and I've been perusing all the articles. I read the FDA papers and then this one, and so personally for myself, but hopefully it helps you guys too. If you guys have not, remember to email Dr. Chen if you are interested in the vaccine. And also, if you have emailed him, just make sure you keep your phones on and keep an eye on it because it's possible the employee health might call for you to go get the vaccine, potentially as early as today. So, yeah, so just keep an eye on that. But thank you so much, everyone. I have recorded this too, so if someone missed it, you can share it with them too. But yeah, thanks, guys.